And welcome back, everyone, for the last session of the day. And this session will be about sensory systems, which should be super fun. I'm your chair. I'm Jenny Voss. I'm a postdoc at the MBL in Woods Hole with Josh Rosenthal, where we study RNA editing in squid and how that may be involved in environmental acclimation. We've studied temperature. Recently, we added oxygen, thanks to a Kavli grant. And that's a lot of fun, and I'd be happy to chat about that later. Now, we are talking about cool sensory systems, acclimations, adaptations in a variety of animals. It's really fun. We've got almost all 5 senses. We've got olfactory things. We've got taste by touch. We've got acoustic things. So we've got almost all 5 senses. We've got fish and moths and bats and octopuses, invertebrates, vertebrates, things in the sea, things in the air. We've got all the things. And the first speaker is Mar Huertas from Texas State University. And Mar studies various fishes. Among these, the goldfish is especially interesting, she thinks, because goldfish can do super many cool things, including communicating with each other. They're super resilient. You can flush them down the toilet, and they're still kind of fine. They can even drive cars, and I believe goldfish will also be featured at least a little bit in this talk. Thank you for speaking.
Because what I'm saying here is not the same.
What a great question.
Okay. Yeah, what I was saying first. Maybe
Can you pull this thing up?
in the display.
At least 4.
Yes. Sorry about that. Well, I can introduce myself. So I'm a so it's kind of a surprise for me, every slide I'm passing. So I'm a comparative physiologist. I work with the sense of smell in fish. And today I'm going to speak about... You cannot hear? Can you hear me now? Oh, they removed the microphone too. Oh, should I introduce myself again? Well, let's go to the subject today. I'm going to speak about also a very interesting subject related with changing ecosystems, that it's the presence of nitrogen compounds in aquatic ecosystems. And now I'm kind of stuck here. We can just go to the normal slide and just...
Because it's still on presenter mode, can you try
Where?
Okay, so
Grape. The mode. This one? Aha! Oh, fantastic. Okay, so finally the first slide. So this presentation is going to be focused in the effect of nitrite in the sense of a smell. And the first thing I'm going to speak is about where does nitrate come from? So nitrogenous compounds are in water from almost the beginning of times. We have accumulation of ammonium from the decomposition of plant and animal matter, and also increases of ammonium due to excretion of compounds in fish. This ammonium is transformed to nitrites and nitrates by the nitrifying bacteria. And the most common form of nitrogenous compounds in water is nitrite. It generally is in the level of nanomolar, micromolar. But in the last decades, with the increased demand of food in the world, we have also increase of agricultural activity and also increase of nitrate-based fertilizers that leads to a runoff of ammonia and an increase of these nitrogenous compounds. And only in the US, we can see that in these areas where agricultural production is more intense, we can see more nitrogenous compounds. Not only this, we have actually set what will be like a safe level of nitrates in water. It's around 10 milligrams per liter. But we are terrible at keeping these levels. Actually, well, you can see here, Texas, where I live, California is always going over these levels. Actually, I'm measuring the nitrate concentration in the fountain in front of my lab, and we're drinking a bunch of nitrate. It's always over 10. And this is where, speaking about first world, you can imagine this in other places where the control of nitrogenous compounds is not as tight. So one might think, we know that ammonia is very toxic, nitrite is very toxic, and nitrate is less toxic. So we were thinking for a while, says, well, it doesn't matter how much nitrate we have in the water. But in the last years, we found that nitrate can be transformed to nitrite in the digestive systems in humans. So if we have an increase of nitrite in our diet, either by drinking water in the fountain in my research center, or in Texas, if you eat barbecue, you can also increase this amount of nitrites. You can increase also these nitrite compounds. And that can lead to a lot of problems. The most interesting thing is it was not until 2021 that we have our first kind of call of attention, says, hey guys, we have a problem with nitrogenous compounds in the US. Just saying that we have an increase of ingestion of nitrogenous compounds that can alter several physiological functions in humans. And at that time I was thinking, well, if this is for humans, imagine for waters that are not treated for human consumption. The amount of nitrogenous compounds that we have in our natural waters is actually staggering. And as a fish physiologist that work with olfaction, I'm thinking in this super sensitive system, these neurons that are projecting their cilia, their sensitive cilia to the water, and are continuously exposed to these nitrogenous compounds. I started my research years ago in aquaculture, so I knew that nitrite is actually a foil in fish cultures. So when it's accumulated in huge, in big concentrations, we're speaking about 2 to 10 millimolar, it leads to a lot of problems like methemoglobinemia, that it's the oxidation, the irreversible oxidation of hemoglobin. There's also other damages like hair damage, ionic imbalance. It's also a chemical irritant, and also leads some problems with lysosomal and liver mitochondrial alterations. Even it's also related that nitrite can induce some cases of cancer. What I was thinking about this new nitrate concentrations is how this chronic exposure at lower concentrations, not at the level of millimolar, 10 millimolar, but micromolar, nanomolar, is affecting the sense of smell. And the last years we've been investigating all these processes. And what I found is that nitrite has like a double-edged sword effect that we already knew for digestive processes in humans or in other physiological process related with nitrite, but not for olfaction. So I'm going to just, in my presentation, I'm going to show you several of these positive and negative processes. The first one is that nitrite can be accumulated at very low concentrations after chronic exposure. And you can see here, you expose fish to 1 millimolar. This is the 10% of the LC50, the lethal concentration that kills 50% of the individuals. It's a 10% also exposed to 1% and 100 times more diluted nitrite. You can see that gill, liver, and kidney is not changing their concentration of nitrite, but the nose and the brain are actually showing an increased concentration at the lowest treatment, the lowest concentration treatments. And that's very, very interesting. So the nose, it seems that it's kind of acting like a portal of entry of nitrite. And it seems that must be some kind of adaptation to uptake this nitrite for the environment. How is this related with function? What you can see here, it's a confocal image of a olfactory rosette of this fish. Let me go back here a moment. This is a dissection of a goldfish brain and olfactory organ. The goldfish is a very, very interesting system. Can you see my cursor? No, let me go here. The brain of the goldfish is very interesting because the olfactory bulb is actually detached. These two little things are the olfactory bulb. It's detached from the main brain, and it's almost located close to the olfactory epithelia. And the olfactory epithelia is this gorgeous rosetta full of lamellas that are very, very sensitive to odorants. If you would see on the confocal, this is a immunostain for cilia. And you can see that in the goldfish, olfactory cilia are distributed in these kind of turfs of sensitive cells. When we expose fish to 0.01 millimolar, this is a very, very low concentration. Goldfish are fine, they eat well, they don't have any of these acute symptoms of exposure to nitrite, but we can see an increase of this surface of cilia. And how is this related to function? We expose these fish to food odorants, and we did this to choice maze analysis, in which we put the goldfish at the end of a maze and let them to decide if they want to go to a food queue or choose water. And you can see here, we calculate the index of preference, and when it's positive, significantly positive means that the fish is attracted to food. You can see here that control fish, of course, they're attracted to pellet food, but fish that are exposed to 0.01 are actually more attracted. It seems like they can detect with more sensitivity these olfactory cues. Then in 0.1, they are still affected, and this, at 1 millimolar, that it's still a low concentration, it's affecting the detection of food. So you can see this double-edged sword effect in which you have low concentrations, it seems that it's a positive effect, but at larger concentrations will have a negative effect. And we found this in several fish species. This is the case of the catfish. We also expose them to several concentrations of nitrite. In this case, it will be 0.2 molar. This is at 10% of the LC50. And you can see here that fish exposed to nitrite have a more sensitive responses, or even as similar as control, to main odorants like bile acids and amino acids. So that's very, very interesting. And the next question is, why are fish more sensitive to odorants when they're exposed to nitrites? And the first thing is, why is this accumulation of nitrite helping with the olfactory process? And for that, first we have to understand how nitrite is incorporated in the nervous system. For the last 3 decades, I think from the 90s, we keep saying, people in aquaculture, we keep saying that nitrite enters through the gill using these anion exchangers that is in charge of osmoregulatory function. So it uptakes chloride from the outside and excretes carbonate outside. And we keep saying that this is the mechanism of entrance of nitrite, and I always believed that. Until going back in bibliography, I found that we keep citing each other, physiologist after physiologist, we keep saying, this is a transporter for nitrite. And no one demonstrated that this is true. The only thing we know is that when we have chloride outside, we have less transport of nitrite, and that's it. And maybe some studies like this is myself, just finding that this transporter is more expressed when we have a treatment with nitrite. So our next step is answer this question, how is nitrite transported? And we express the anion exchangers in Xenopus oocytes, and we measure either electrogenic current or transport of nitrite, accumulation of nitrite inside the oocytes. And this has been like a year an endeavor, and this is the only results we found until now. That happened the second day we injected oocytes and never again. So this is how electrophysiology works sometimes. And we still don't understand why we can find these electrogenic signals. We can see here in chloride, this is the one that we know for this gene. But what is happening with nitrite? We believe that temperature can actually alter this transport, because at that time in the lab, we have some problems with heating. So half of the corridor was at 30 degrees, and half of the corridor was like 10 degrees. So just going between labs, we have a big change of temperature. So we're still working on that. But when we check the increase of concentration inside these oocytes, we also found that at 1 millimolar, we can see a concentration of nitrite inside the oocytes, but at 3, it's actually going down. And the exposure to chloride and nitrate together is not affecting this transport. So there is still a lot to explore in these nitrite transporters. Actually, it's not only fish that we have a very low, I have no idea what is happening. Also in humans, we still don't know how these nitrates and nitrites are going to be incorporated inside the organism. The second question, the second gap is what happened with nitrite, what it's inside? Why we have this increase in olfactory function? And this is what we propose. We know that nitrite can be transformed to nitric oxide through the non-canonical pathway of production of NO. So we think that fish have also this ability to produce nitric oxide when they have an increase of a nitrite concentration. So until now, we measure increases of nitric oxide in hemoglobin of fish exposed to nitrite, but it's still a story to be developed. So this is a big question that I think is going to help to understand how changes in our ecosystems are going to affect the chemical sense of fish. Because if we have fish that are exposed to different environments with more or less oxygen, these non-canonical pathways that are non-dependent of oxygen are going to be more important. That means that fish that have no access to oxygen, they can still make their chemical senses work, and even their brains, by using this pathway. So I'm going to go very fast about the negative effects of nitrite in chemical senses. I'm going back to this beautiful rosetta that fish have and how they have all the distribution of these olfactory sensory neurons. And just to show you here how it looks in an olfactory epithelia of a catfish that is exposed for 30 days to nitrite. This is a control olfactory epithelia. You can see here the different olfactory sensory neurons. And these electric blue dots are actually mucus cells. And after 30 days of nitrite exposure, you can see here an increase of this mucus layer and also infiltration of different immune cells. So what it feels is like an immune response, an inflammatory response to continuous exposure to nitrite. Again, this fish looks fine. It's eating, it's not showing any kind of stress, but the nose is kind of stuffed. Another effect that we see in the exposure of nitrites, it's the microbiome in the nose. So the microbiome is dramatically affected by exposures to nitrite at very low concentrations. Just to explain a little bit about this figure, this is the composition, every column is the composition of the microbiome of one single fish nose. You can see every color represents a different genus. What it's easy to see is that the appearance and disappearance of genus between different nitrite concentrations. So nitrite dramatically changed this makeup of microbiome. And the microbiome is also going to be related with the transduction of signals in the olfactory epithelia. So a lot of things are going on. First, this kind of inflammatory process, the environment, the microbiology environment is changing, and other things are happening also in terms of behavior. This is an experiment in which we expose several fish to several concentrations of nitrite, just I think 2 hours post-hatching. And this heat map, what it's showing is movement of larvae after exposure to nitrite. The more clear color, yellow, white, means less movement, and the darker color means more movement. And you can see that there is a tendency of increase of movement with increase of nitrate concentrations until the point that when you reach 1 millimolar, this is close to the LC50 fish just stop. So you can see a significant increase of movement just by exposure of nitrite. What are the applications of all these new experiments, of all this data? How can we integrate changes in the environment and how it's affecting vertebrates, not only fish, but also humans? So we proposed a while ago to study how nitrites, ingestion of nitrites, this nitrite that actually I told you that we can find in our own waters, how can affect one of the most sensitive sectors of our population, that it's pregnant women. But obviously, I cannot take pregnant women and just give them nitrites and see what happened. So fish are a fantastic model to study these effects, because we have species that can get pregnant. And this species can also be exposed to nitrite and see how this affects alters neural development. And also use olfaction to determine how this alteration of the nervous system is made by nitrite. So we started to do this project like a year ago. We were not very lucky in terms that we exposed to nitrite these pregnant mothers, and they just didn't give birth. So it seems that nitrite also alters the development of the embryo and even the fecundity of the mother. But we are starting to do new experiments at lower concentrations and also at later stages of sex maturation. And we are having our first youngs, and we are going to see how this exposure to nitrite is affecting neural development and olfactory processes. So all this to say that fish have a very sensitive nose. And they are using these chemical signals to interact with the environment to coordinate very important physiological functions like reproduction, imprinting, migration. And just highlight here the ones that we study in my lab, that it's reproduction, food detection, and the sensing of pathogens. And my students, when they're presenting these results in conference, they like to say, nitrite is a silent killer. And this is because when we tackle with the chemical senses, with the sense of a smell, we're actually stopping this signal. Imagine this is a button that it's activating a computer. So when we eliminate the ability to sense chemicals, when the sense of smell disappears, these processes, reproduction, food detection, are also affected. Does it kill the fish immediately? No. But in the long term, it can affect the survival of a species. So that's why it's so important to understand how nitrites are affecting the chemical senses of fish. So this one is a slide that tried a little bit to answer the questions that were requested to us about identifying the main gaps that we feel that are important for researching and changing ecosystems. And from my point of view, I think the story of nitric oxide in the chemical senses is clearly unexplored, much more in aquatic organisms. And they can give us a lot of information on what is going on in these responses to changing ecosystems. And also, we need to identify transporters of all these toxicants, not only nitrites, but any kind of toxicological compound. We need to identify how are incorporated in organism. So what are the other things in the future that I would like to see? I would like to actually see more a link between this omics, this huge integration and production of big sets amount of data. I would like to see how is this going to be translated to the function of the individual and populations. And it seems that the more we know about expression of genes and mapping neural circuits in animals, the less we can understand how is this translated to function. So for me, this chiasm between genomics and function is getting bigger. And then the other thing is also to overcome this chiasm between the different areas of research in biology. It's necessary to create these highly integrated and integrated research systems. And I'm not saying anything new. We're saying this for decades. And I think it's still important and sensitive to do this kind of meetings in which we are sharing very diverse information and different techniques and different models and different points of view on how changing ecosystems are affecting individuals. So with that, I like to thank my collaborators and my students at Texas State and the different funding for NIH, NSF, and the USDA. And with that, if you have any questions.
Thank you. Any questions? Yeah, we got one right there.
Thank you very much. I found the result with the nitrite with low, having better detection to low concentrations very interesting. And I was curious about how, kind of what different stimuli you might have presented to kind of see, is it just in relation to food odors? Have you looked into individual amino acids such as alanine, which is very attractive, or even gone into further with such like repulsive components such as cadaverin that was brought up earlier, or pheromones even. So I was curious.
Yeah, the compounds that I found doing electrophytograms are amino acid, bile acids, and pheromones. So that are the main compounds we work in my lab. I didn't work with any alarm cue or espermin or putrescine. I didn't thought about that, but the main sensitive odorants in fish have been tested, and all of them have an increase in sensitivity. Bile acids are, I think, they're more affected in this process. They are more sensitive, which is very interesting because these are the signals that are more related with chemical communication and pheromone sensing.
All right, I think we have time for one more question. Great, me. I was really curious to see that the microbiome shifted. Do you actually see inflammation or infections happening in these animals, or have you left them long enough or challenged them maybe even with a pathogenic
That's
bacteria?
a very good question. Actually, we did this with catfish, and we did four treatments, one with nitrite, one with probiotics, and one probiotics and nitrite. And if you alter the microbiome with probiotics, they are less sensitive to pathogen infection for neurotropic viruses.
That's very
So,
cool.
yeah,
Awesome. Thank
yeah.
you so much.